Hello guys, and in this video we'll see about the BSc syllabus, that is zoology third year. So here in zoology third year we have to study deep study of three topics, that is physiology, genetics and molecular biology. Let's see the full marks here given 100 and pass marks 35, so third year. So let's talk, that is a first group, that is physiology, second genetics and third one is molecular biology. So first one is our physiology. So what should we study in physiology? That is 75 lecture hour. So first topics is nutritive substance, enzyme and energetic. So in this topics we will study biological role of nutritive substance, metabolism and metabolic pathway, energetics, bioenergetic and general properties. Okay, so then after here, and the mechanism of action of enzyme, cofactor and coenzyme factor influencing enzyme activity. So then again topics, that is in physiology, that is digestion. So in digestion what we study, digestion and absorption of nutrient, gastrointestinal secretion and its regulation. Then after respiration, so in respiration what we study, that is respiratory mechanism, respiratory pigments and respiratory gases, regulation of respiration. Then after in circulation, that is blood groups, homeostasis and blood coagulation, conduction system of the heart, cardiac output and its control. And then in excretion and excretory products in animal, mechanism of urine formation, role of kidney in maintenance of electrolyte balance and pH. Then after here nervous systems. So in nervous systems, nerve cell, electrical signaling, synaptic transmission and neural integration. Then after endocrine gland, so here we study primary endocrine glands types. Then after respective hormones and the function of these hormones secreted by endocrine glands. And then after sensory systems, so general principle of sensory physiology, that is vision, hearing and balance, taste, smell and touch. And then last one is our reproduction and development. So in this topics what we study, that is female reproductive cycle, it means ovarian and uterine cycle in human, gametogenesis, types of eggs, mechanism of fertilization, embryonic development. So this is the first, that is physiology. And then after our group B, that is cell biology and genetics and molecular biology. So this one is total first, that is B1, that is divided into two, that is B1 and B2 and side B2. So B1 cell biology, that is 22 lecture hour. So let's see what we study in, that is cell biology. So first one is general organization of prokaryotes and eukaryote cells. Then after cell membrane and molecular organization, membrane transport principle, cytoskeleton and cell motility, microtubules, microfilaments, intermediate filament, cilia and flagella. Then after structure and function of endoplasmic reticulum, Golgi complex, lysosomes, perioxysomes, mitochondria and ribosomes. Then after nucleus, structure of nuclear envelope, nucleoplasm, chromatin fiber and nucleolus, nucleocytoplasmic interrelationship. Then after here chromosomes and chromatin, we study nomenclature, karyotype, gained chromosome, heterochromatin and euchromatin. Then after last one from here, cell cycle and cell division. So cell cycle, mitosis and meiosis, cell division. And B2 is our, that is genetics. So what we study in genetics, let's see one by one. That is in genetics, first one is Mendelian and non-Mendelian inheritance, that is 2 lecture hours. And genetic interaction, gene alleles, dominant and recessive, multiple alleles, blood group of human, and linkage and crossing over theory types of significance, sex link inheritance, characteristics of XX and YXY link gene inheritance, non disjoint as proof of chromosomal basis of heredity, and then after sex determination in animals, then after chromosomal variation, chromosomal abbreviation, euploidy, monoploidy, polyploidy, nullsomy, trisomy, double trisomy, tetrasomy, mutation and their types, human genetics, pedigree analysis, human traits, sex linked disease, disorder due to mutant genes, eugenics, euphenics, and last one, genetic engineering and gene therapy, that is introduction and their application. And last topics, that is what we study in molecular biology, that is 34 lecture hour. So in molecular biology, that is first one is our nucleic acids, so structure and composition of DNA replication, DNA polymerize, polymerization properties and mechanism of action, semi discontinuous, unidirectional and bidirectional DNA replication mechanism in prokaryotes and eukaryotes, structure and composition of DNA, RNA processing of messenger RNA, ribosomal RNA, and transfer RNA. That is then after genetic code and central dogma, characteristics and oval hypothesis, concept of central dogma, and here last one, that is transcription, translation, and protein synthesis. So difference between replication and transcription, RNA polymerase in prokaryotes, properties and organization of promoter, mechanism of prokaryotic and eukaryotic transcription, mechanism of translation, initiation, elongation, termination, translation process in prokaryotes and eukaryotes, post modification of released proteins, and gene regulation in that topic, gene expression, regulation and control in prokaryotes and eukaryotes, translational and post translational modification system control at hormonal level, technique for molecular biology.